you the reality so that you know and understand that the challenge ahead of us and how many of the projects and ideas are even far more important. We will do them anyway. These are important problems to work on, but I will give you a little bit of a flavor of behind the scenes, what's going on. And since I'm just thinking about this is what we should do, I'll pull out some decks, but also we'll do this as a conversation. Let's connect the projector.
Yeah, so do you have an HDMI port?
I have that. So if I move this here, let's do this. We will do what time, how long do people have today? Because I know we started half an hour late. So if we go on, we were supposed to go on till 12:20, and instead we could still just go on till 12:20. So we have a half, maybe 50 minutes. I'll do half an hour and then you'll do 20 minutes on Shishto. And I think instead of what we are doing, spend a lot more time on the problem itself. Because that might be, I think the purpose here is to just get you all to understand a context of a problem. I'm curious, how many of you are thinking about global health as a problem to pick on, or infectious diseases in general? So at least a few of you. It's worth sharing. For the last 10 years, we've been working on malaria diagnostics. I want to scare you a little bit. That's my only job in this class. It's for every problem. If it's going to be oceans, I want to scare you a little bit. Not too much that you say, oh, it's done, but enough that you can't sleep tonight. That's the goal. Okay. So if I don't succeed, tell me. Or if I do succeed, also let me know that you were sleeping and thinking about what we talked about today. That's my only goal. Okay, we can record.
Yeah, I'm recording right
now. Shit. Okay, you did record that. That's good. Yeah, no, I think for the class, and we are just about to send the flyers today. I already started sending and signups are coming in. So by Friday, we would finalize the online community itself that will be joining, including many of Will's team members and other factors itself. And I think this is a fun example of just, there is sort of an organic nature to this class, driven much more by the fact that, oh, do I need to, I see. Yeah, you're right. Yeah. You are right. Before I do that, let me do certain sets of things here. What do I care about? Okay, so how do I join on Zoom?
Just on Slack in the Ian and me channel.
Got it. Kevin. Okay, before I forget, there's a really fun session on campus today starting at 12:30. It's called Measuring the Pulse of the Planet. I don't know if you have to register or not, but there's some, if people care about oceans and measurement technologies, you should all come to that session. We'll spend some time talking about measurements in the ocean, but people are talking about methane measurements and all kinds of other measurements. Kevin and Ian, if you guys can find that PDF that I had sent and just send it to a class channel, that might actually be useful. Okay, and then let's, okay, so how do I do this now? Recording. Here. Yeah. Okay, perfect. What do I want to say about malaria? So let's start with how many of you have gotten malaria? Show of hands. You think you might have got, say that again? Dengue, yes. Malaria, you don't know. It's also possible that you've gotten malaria and you fought it. How many of you grew outside US? Just a show of hands, at least a few of you. Um, so I think, I mean, we will broadly talk about mosquito-borne diseases. I think the purpose of this discussion is much more about the complexity when diseases are transmitted in the sets of ecological sets of frameworks. Mosquito happens to be, and I think in schistosomiasis, you will see a parallel side of the story. So often when we think about solutions, we are so gravitated towards the pharma. And of course, those are necessary. You got to treat people. But there is a broader aspect to it, which helps scenario around how environmentally entrained many of these sets of diseases are. So first thread on, this is a little bit of a, for California, for example, I mean, clearly we had malaria in the past in United States. That's been known for the longest time. Some of you might have seen, how many of you have seen an ad from Disney about malaria? You guys know this history? Okay, I think we have to start the fun way then. If you have not seen this, then I have to now start by, we did this in our science, we do art and science in our lab every few months in a group meeting. Disney film Malaria. So nobody has seen The Winged Scrooge at all. Okay, I won't play the whole film because it'll take too much time. I'd like you all to see The Winged Scrooge. 1940s, the military was struggling. That's why it's a little bit ironic to show this film because this was funded by the US military per se, because of course, and now it's a complete reversal of what we think about the role of military and other sets of factors are. But just the background is, was hired because they were realizing that the efforts in elimination of malaria were very limited, and they wanted to essentially talk about malaria in an interesting way that would capture people's attention. So I'm just going to play this, and the purpose of this is not the information. We'll gather some of the information through the stats, but I love the idea that it was the cutting edge of animation at that time. Of science and the challenge of a disease, but in a very playful way, which is a little bit of a surprise at the time. So let's just watch 2 minutes of this. They're American affairs. I don't know if those affairs exist anymore. That's how it used to be.
Public enemy number one, Anopheles mosquito, wanted for willful spreading of disease and theft of working hours, for bringing sickness and misery to untold millions in many parts of the world.
You can see the U.S.
The tiny criminal is linked to the destinies of man in a cycle of disease transmission that could not exist without either man or mosquito. Each is solely dependent on the other for the existence of the dread malaria. Is readily distinguished from other mosquitoes by the fact that she stands on her head at an angle of 45 degrees or more.
I don't know if you guys have noticed that.
This newly hatched female has not yet touched human blood, therefore does not contain malaria. But if we follow her, we'll soon learn how she becomes a malaria carrier. Like all thieves and killers, she works best under cover of darkness. Locking and barring windows will not keep this hungry prowler out. She can enter through a small crack. What will she find to steal here? Only a little blood, which this man, racked with the chills and fever of malaria, will never miss. Assuming her typical angle of 45 degrees or more, she injects saliva to make penetration easier. And in a few seconds, she begins. But this man is sick. Along with his blood, she's drinking in the parasites which cause malaria. Gorged with disease-laden blood, she makes her way to some cool, dark place where she will rest for several days and digest the blood. Oh,
So I'm going to stop here. I think one of the threads there is a scenario for you to understand that communication in some of these threads is incredibly important in how you relate to the public per se, and what are current dangers to a disease. So now I'm going to ask you a question. Do we still have Anopheles in California? Right? I mean, we don't hear often, you're hearing about measles being present in the country, but do you know, could we still spread malaria in California? How many of you think yes? How many of you think no, we've pushed that far away now? Yes, yes. So it turns out we actually do have Anopheles in the mountains, and malaria can transmit in many parts of the US still. But we control that by treating people. And so effectively, when the numbers go down, you don't get transmission cycles. When you don't get this is a classic case in infectious diseases. Below a certain number, they will die out. Above a certain number, that they pile up. Sort of my story of thinking about malaria sort of begins with this photograph. This is from 2013. I'd been working on malaria for a year and a half. And majority of that early on was really trying to understand the sets of questions. There is a remarkable number of scientific questions that remain open. But it was important to just also understand the ground reality feels like. And so this is a photograph from Uganda. And one of the important, I mean, there are many things that you can see in this photograph. You see a pregnant mother in the back, you see a lot of kids playing, but something that you only realize when I was walking away. This is one of our field sites in Tororo. Tororo is at the border town between Uganda and Kenya. Jesse, who's back from there, UCSF has a malaria eliminate program there. So it's a massive program. Tororo is a very important site. But I don't know if you recognize that these kids are playing on those little tables are actually, it's a graveyard. So that's the dead from the family. And in some sense, there is a tradition and you drive and you start seeing this pattern everywhere. People bury their dead because they don't want to forget. Most people also don't name their children often enough early, because for the first 5 years, you really don't know who's going to make it. And this is the ground reality. And you might think, oh, 2013, okay, we might have done better, right? So 2013, we spent as a community, every year, roughly the budget for malaria from the President's Malaria Fund was one of the important USAID, which we will talk a little bit about. There is the Global Fund, roughly between 3 to 4 billion years. $3 to $4 billion are spent per year on malaria and control of malaria. I'll let you guess. The numbers, let's just first think about numbers from an number of infections across the world. What might be a number that you can guess that we had for, say, in 2011, some number like that? What number comes to mind if you were to ask? I mean, let me back that. It's known, and it's been continuous over the years, roughly 500,000 kids die of malaria every year. That's the reality of the current sets of, but it's thinking a little bit about total number of infections that gives you how big a problem this is. So anybody wants to guess a number? Yeah.
40 million.
20 million. Okay, that's a good number. Anybody else? Everybody's order. Yeah. 50 million order of magnitude. Okay, that's a little bit higher. That's a lot of people, right? I mean, what is the population of California? Similar order of magnitude, right? Okay, we will come back to that. How many of you believe that since 2013 to now 2023, 2025, has the number gone up or the number has gone down? I mean, we have spent $4 billion every year, year after year. Yeah. Down. That would be the very rational thing to say. And the reason I don't sleep at night is this, that in fact, this is a chart. 2011, the numbers roughly were around, if you look in terms of, this is 229 million. There has been a steady decline in malaria for 50 years. So if you were to look at this chart 50 years back, this chart would have been astronomical, and we have consistently making progress. Majority of this comes from bed nets. And then after that, you start seeing a trend. And at this point, when you look at 2025 numbers, for example, that number is 263 million. So something is not right. Like if you look at this, and this is with all the, I have not gotten to any statement around support has changed. I'm not talking about what has happened in the last couple of months. So you can just ask yourself a question of, first of all, we have to think about this because something is not right, and maybe the low-hanging fruits are gone. Maybe the sets of things that were working are not working. Maybe we are better at diagnosing. Maybe these numbers were just large enough to begin. The diagnostics programs are getting better, and that actually is a viable explanation. But at the end of the day, it's important to kind of recognize the fact that we don't have this under control by any means or forms. That's kind of the first thing that comes to mind when you think about these sets of numbers. The other part of this story is, in terms of the number of deaths has been fairly steady. But there are two other that I really worry about when I think about malaria per se. And then I'll transition and tell you some backstories about what's happening. So I don't know, some of you have heard artemisinin resistance. So this is the frontline drug. We got a Nobel Prize for that. That was discovered through Chinese medicine. So again, going back to that perspective, if you think about traditional medicine and its role, remarkable intervention that was made. People actually understood the biochemistry, isolated the molecule. That is the frontline drug currently. Cambodia, Thailand, Indonesia, essentially a resistant emerge for this. And now that resistance has spread across Africa. There are lots of drugs in the pipeline per se, but I would say there is nothing close to actually being deployed as yet. So there is a big gap between what, if something, if we have a big jump currently, there's not that we're going to do something about the resistance that's been already reported. There is a massive amount of now capability because of sequencing to be able to detect this resistance per se. So now if this was not enough, there is something else happened, which by itself is really interesting, which do with RDTs. So we will talk quite a lot about RDTs and many of these kind of an immune assays that are associated with quick, rapid diagnostic tests. But ironically, the gene that was chosen for the disease, which is called the HRP23, PF stands for Plasmodium falciparum. We'll talk a little bit about that. Somehow, and in terms of total number of tests, roughly between 200 to 300 million tests are run around the world. And we ended up selecting a gene that is not essential for the pathogen. And so what does the pathogen do? It actually develops resistance against our frontline diagnostics itself. So currently, we are in this situation, and this is a graph from last year, that across the world, we have detected the widespread resistance to the diagnostic test itself. So the irony in that is that you keep treating people, but you don't treat the people. And so the biggest infection that spreads and leaks out is the one that's resistant. So by actively only treating people, which is the WHO standard, you end up in a situation that this is very unique. It doesn't happen that often because people have to be careful. And I would love for somebody to dig into the history of this, that when they chose this gene, why did nobody ask the question whether we could develop resistance against it or not? So it would be a useful question in how we make decisions around, especially if you're, so much of the funds actually support RDTs. Roughly RDTs in the current clinics can cost between $2 to $5 per test. So that's an astronomical price if you were to think about it, but are subsidized primarily because of the kinds of care that's been built in some sense. And by the way, anybody should interrupt anytime. This is a random walk into the world of malaria. I'm just trying to tell you why I don't sleep when I think about malaria. It's been now 12, 13 years we've been working on this problem, and the problem only keeps growing in some sense. So, at that time, we were not sure. We were seeing this set of a spread of the resistance more and more. Then there is a third challenge, which actually is probably one of the biggest one, which happens to be what is called a super mosquito. How do I hide this? Okay. So we just talked about Anopheles. There are many species of Anopheles. Anopheles gambiae is the one that transmits falciparum in Africa. But there is a new mosquito that's been reported recently. So this started around 5, 6 years ago. But some of the latest infections that occur or large-scale spread events occurred because of a different mosquito, which is Stephensi. So this is being called the super mosquito primarily because it thrives in urban environments. So if you were to think about that picture and the photograph that we was a rural setting, population densities are low, and so disease doesn't spread so rapidly. While Stephensi loves living in cities. So as a mosquito, it spread over across from India. And one of the ideas is that it was actually around Horn of Africa through trading. People started reporting it here. So this began, and now with the latest sets of challenges in Ethiopia, there is widespread spread of a new mosquito that spreads this disease. Now, why should this worrisome? This is worrisome because if you think about Lagos and you think of many of these big cities, you have very high population densities. So we, for the first time, have a vector capable of surviving in urban environment. So unlike Anopheles gambiae, which doesn't survive any environments, doesn't survive the dry heat, it doesn't survive small ponds of water, it needs large lakes. Stephensi can live in a bottle cap, a tiny drop of water in a bottle cap, or in tiny trunk holes, can survive in the coolers and just very urban environments. So, one of the things that a lot of people have been talking about for some time right now, this notion of this, that there is a trifecta of these sets of things happening. And in the last, I would say, 3 months per se, then we are hit with this. So, in malaria, there has been this idea of hesitation on using the E word. So E words in global health communities are E words stand for eliminate. When is it okay? And I'm actually curious, how do you all think for us to use that word? Elimination is really about eradicating a disease. Polio, we almost have, but there are pockets of the world where still polio keeps coming back. Eliminate as a word, it's a little bit tricky because if you say it prematurely, we are so far away from any of the targets. So if you were to talk about eliminate currently in the context of malaria, which is being talked about, we are so far away. I think it's been a country-specific approach has played out. specific countries every year that have low transmission try to work very hard to get to elimination status. It's a very proud thing. So India has declared they're going to eliminate malaria. 2030 was one of the goals. I don't see that happening as yet. But at that same time, there has been this notion of thinking about people's commitment towards the disease per se. Out of that 3+ billion that gets invested, there has been a program since the 1970s that's called the President's Malaria Initiative, which pitches in roughly around $1 billion. That was going through USAID, now is completely, USAID doesn't have a website, and we can do this live. I wonder if PMI as a program has a website. So let's do President Malaria in between 30 to 35% of all malaria funding. And actually, the UK commitment also is in that same, so pmi.gov. And this website is undergoing maintenance as we expeditiously and thoroughly review all. So the fact that the website doesn't exist, forget about, it's not about the funding doesn't exist. And again, born down to many of the sets of people that have been working in this space, and specifically, I'm only talking about malaria. I'll talk about impact on other sets of global health sets of diseases. There's lots of issues around trust and trust in the fact that are we actually serious about these sets of diseases. Malaria has complications around the fact that falciparum is deadly. So if you, which most kids don't, you between 10 to 15 days is what you have from the moment that you diagnose a person to be able to treat them. And falciparum is actually, you die. Vivax, other sets of strains, they don't kill. They have a huge impact. And I think this is something that people have understood, that Vivax as a disease also has a huge impact, DALI, and has sets of challenges, but has not been to an extent, people have not thought about Vivax as carefully as possible. Currently, there was a massive amount of momentum. I would say until December, we ran the last malaria workshop in Senegal, teaching and training. There was a lot of momentum that we will make vaccines work. The last WHO vaccines that were declared, they have very low efficacy, 15%. It's difficult to say to give these vaccines out at a large scale when the rollout and kind of the benefits are low. They also have to be given very regularly. NIH has been one of the frontier organizations that works on malaria vaccines. So I think that's definitely something that I directly know as a work moving forward is challenging. And I think, in some sense, one of the ways that you think about, start sort of realizing that we have this multi-prong, and malaria, it's not the first time. Politics has always been one of the key reasons malaria has been very hard to eliminate, or most war zones, malaria comes back pretty quickly. So when sets of things happen in Sudan, whenever there is uncertainty, there is unchecked disease. And this is a very common, people have really documented very well political uncertainty and associated aspects. So I think one of the threads around this, and I'll give you an example of just what malaria diagnostics look like. So this is Banda Fassi. This is one of our field sites. And I'm wondering, Kevin, do we have, I'm just wondering whether we have a Senegal photos in a deck. Link to that deck for a second on Slack. I'll just show people a little bit of our field site and what the physicality of it looks like. This is one of the field sites in Senegal. Some positive news around malaria. Our last trip to Senegal, it's the same location we visited 5 years in a gap. Every primary health center had solar panels. Super exciting to see. solar, anything that we can do in solar coupled to stuff is a super important thing. At this point, the cost of solar is coming down so dramatically. Ironically, this site is actually in the middle of the jungle. And to an extent, in the past, at least, there were reports of illegal gold mining, and that's why people came there. And this is at the intersection Colombia, Mali, Senegal. Mali has a lot of political issues right now. So a lot of people come. So even if one country has a very good control program, the other sets of countries don't, and you get complete spread. The same scenario is in play in Thailand and Myanmar, because Myanmar, there is no control. Thailand has a very good control program. Actually, some of the best malaria control programs are from Thailand.
You want me to share my screen?
Actually, if you just link, I can download and I can then go through, because I'm not going to spend too much time. I just want to say two more things. So let me just share one word, and I'm just going to end with a little bit of an optimistic note on what we've been working on malaria. This is a platform that we will talk technically quite a lot about, which is automation of microscopy. Among several of the platforms that everybody in the class will have a chance to use, this is one of them. I'll bring that in the next class. Play program, and I'll say a lot more about it, but I just want to give a hint, and then I want to transition for Will to talk a little bit about why schistosomiasis is still a problem that we can't check, so that you get a balanced picture. So what does it take to diagnose somebody of malaria? One was RDTs. The problem is that they have very low sensitivity. The other is microscopy, which has very high sensitivity. So this is one of our field sites in Kalahandi, areas in India. And how many of you do microscopy every day? I'm just curious. Okay, at least 2 or 3 of you. So, if I was to tell you to do 10 hours a day, every single day, 7 days a week, this is what it looks like, nonstop. You can see the window, somebody comes and knocks to say, oh, is my report card ready? The pink slip is basically everybody's record, a health record. Ironically, WHO's recommended method for malaria microscopy is called the GIMSA stain. If some of you are interested, you can actually learn how to do this GIMSA stain. But it was described by Mr. Von Gimsa in 1904. And that is still our gold standard. One of the problems with the stain is that it crystallizes, it forms microcrystals, and microcrystals look exactly. And so you kind of realize like, oh, so of course it stains and it has been used, but so stains have to be made fresh. You have to be really good. And then of course you have to do imaging the entire time. And so one of the things that there are 2 or 3, and this is now kind of referencing to sets of projects. There's a lot of work that we have done on mosquito surveillance, which I'll talk about in the future. Blind. So if I was to say how many Anopheles stephensii are there in Palo Alto, the answer is nobody knows, because we have defunded most of the programs long time ago, actually, for mosquito surveillance. The same is very hard to say unless you understand the transmission of these sets of. So that part is important. From a technological point of view, there is a lot that we can do. The other aspect of it is disease diagnostics, and of course, what we have been building, which is an instrument that I'll just show you all. This is what Kevin and I took out to the field. We call this Octopi. It's an object that big. It cost us roughly around $3,000 to $5,000 to build. And then I'm just gonna do a quick video of it so you get a chance to see it. If some help us assemble, just as a way for you to learn optics, as one of the Friday sessions, we have been shipping these to a lot of field sites. Now 6 or 7 field sites have them. And the idea or the joke, kind of just saying this out loud effectively, is you're all in San Francisco. So I find this dramatically disturbing that every day right outside my house, And every day, I mean, how many of you have taken a Waymo? Some of you. If you have not taken a Waymo, here is my recommendation. Take it. Not that you need it, but you should also just understand culturally what's happening around us. And there's a lot to be said that we can unpack. But one of the big things for us was if this can be done, we should be able to do this. It took us 10 years, starting with actually Jim just walked in when he was the first graduate student in the lab. We started working on this. And at this point, it's an entire, and I'm not going to talk about how it works, but I just want to give you a quick demo on what we were able to build. So that's the machine. And this has now a real-time engine for roughly around 20 million cells image. So we just put that slide. That slide has parasites. It has a 30-second stain, which is DAPI itself. And the reason we're able to do this is we discovered a spectral shift that occurs between when DAPI RNA versus DNA. So we're not looking for morphology alone. It actually is a molecular signature for what is in the cell and what is the ratio of DNA to RNA content. That was the key, that you let chemistry do half the work, you let optics do half the work, and you let computing do half the work. Halves don't add up, but you get the point. So what's happening is this video is real time, and it's essentially picking up parasites in real time. This is trained in the field. When Kevin was there, we actually collected data for roughly around, or we tried to collect data for roughly around 1,000 patients. We couldn't finish all the sets of scans. But at this point, there are 5 or 6 sites that we are rolling this out to demonstrate validation. The first sets of papers are out, so anybody can read it. It's called Octopi 2.0. And one of the things that I'm excited about, and I'm spending a little bit of time on this, and I'll talk about the science behind it at some point of time, is that we are repurposing. So this is really the sets of sites where the instruments are in. That's Ethan in the lab, Deepak, that's Liberia, that's one of the sites in Senegal. These are some of the clinical sites that are now ready. And one of the things that's important about this is the fact that the same instrument programmed to essentially detect and diagnose multiple diseases. So the point around this is, the joke is thinking about an app store for diagnostics. And we're releasing this as a tool. All the software, everything is open. And so anybody who has clinical expertise, like you said, leprosy, for example. So we have had several conversations with groups that have worked in. Another one is, I don't know how many of you know elephantiasis, when you get this massive foot that's swollen. Again, microscopy-based diagnostics. If you have a clinical partner, you train the machine, and suddenly the same machine is capable of running more and more sets of tests. So one of the purposes of this, and the reason I'm just mentioning this, is that this as a platform will be available for anybody who's excited about the intersection of ML and real-time ML, especially disease that, especially thinking about building meta layer tools as well on top of it. Because the thing that's very exciting about this as a platform is, rather than us holding the sets of diagnostic tests, the goal is to release this in the community, and a clinician or a clinical team or a researcher, say a research group in Tunisia, should be able to bring and they have expertise in. So our goal is to build the meta layer tools, like annotation tools, tools to validate, and such that a much larger group of people, so that we are able to bring online. It doesn't take us, it took us 10 years to get to this point to get a tool for malaria. Now, should it take us another 10 years, another 10 years? So the idea is, how do you rapidly bring image-based diagnostics to the market? So this is why I'm we are going to switch context unless somebody has a comment on malaria. Let's take that as a conversation while Will, of course, Will isn't prepared. I hadn't told him that he's going to present something, but join in, connect your computer, and let's talk a little bit about schistosomiasis. And in the meantime, while Will is connecting, any comments? What comes to people's mind when you hear about malaria? Any questions? This is a whirlwind into, yes. Yes. Yeah, do you want to just use this? The short answer is we don't know. We have some instruments out in the field now for almost 2 years. But since the tools keep changing, we have been upgrading them from time to time. And some of the usability studies that we are trying to do now, we have a capability to be able to log in into the instruments. And this is the purpose of deploying them at a much larger scale, is to really see the status of the instruments. How many times were they used? What does the log look like for the set of validity? And then a little bit them from far itself. I think the current design challenge per se, before we do the mass production side of the story, is really around robustness. So when Kevin and I came back from Senegal, I think I roughly calculated, we assembled and disassembled the machine probably roughly 50 times in a period of 15 days. We would go to a clinic, it's packed and disassembled. In the next 15 minutes, right in front of the clinicians, we would put it together. And we would disassemble it again. We would go to another clinic, say 2 hours away. We would put that back together. And then we were just trying to understand what aspects should be modular, what aspects should be. Dust is a huge issue. Dust really kills a lot of optics. And so currently there is a student in the lab and we are borrowing a dust filter built for the World War I. In the Jeeps, there is a type of an oil filter, a gravity-based oil filter. So, I mean, desktop computers survive in these conditions. So if a desktop can survive, we should be able to have this survive. But it's not a given as yet. But the goal really is that the idea is that these machines should have a lifetime of 5 years as a test case of the target. This is not an oil objective. These are all air objectives. And the part is the stage itself. I think there is another layer of design in which we can reduce the number of moving parts also. But the only way we'll find out, I think so far we've built around 6 or 7 of these that are spread across. But because we keep changing the design, it's unfair to say that, you know, we've taken out stuff that was printed, 3D printed when we went to Madagascar, and that didn't survive. That's why you see everything is machined. And so there's a lot of less. Okay, any other comments on malaria?
I wanted to comment.
Yes.
I learned something today about the Stevensi
Yes.
that's being seen to be more in the cities, because I didn't realize, it's really concerning that there's little tiny pools of water that these mosquitoes can proliferate in. Especially, like, I was just learning about remote sensing in the Amazon to look at stagnant water and high flow water and try to map with drones where the mosquitoes are. And that's a very big,
None of that works.
that's not going to work in the city. Like, this is, the resolution for like using satellites, for instance, or for using drones, we're talking about centimeters and sometimes meters. And then if you have a little tiny pool of water. So anyway, that's just really concerning, because basically all the strategies, even like the most cutting edge strategies, are really be able to solve that. So,
Yeah, I think urban mosquitoes are a huge problem.
yeah.
And one way of thinking about it is that urban mosquitoes present themselves in ways and forms that there is a big difference between vector control in rural areas. And again, there's also sensitivity around blunt approaches for mosquito control. So I was a kid, DDT spray was a common, and I don't know how many of you remember this, but we thought it was had some white thing coming out, and as kids we would run behind the truck. It's like thinking it's some mist. Now I know what the hell we were doing, and it would not be okay now. But many countries eliminated malaria in mosquitoes primarily by killing everything. And just later on, we actually learned it's still DDT, you know, it's one of the most effective things, but it's not sufficient. And we can talk a lot about the biology side of that story.
Wow.
Yes.
One question on malaria, the lab that I do for research with on climate change.
This is
Modified lab.
yes,
So
yes, they do a lot of modeling.
Malaria and other vector-borne diseases as well.
Yeah.
And so I think climate is also much factor in this because obviously malaria has been surging in Peru and other Latin American countries where it historically hasn't.
Yeah.
And that's a huge emerging trend because the resource, like in India, we do have malaria season, but we know get your nets out, be careful, like.
And also there is expertise to handle a disease as well too.
Yeah.
Yeah.
And
I think the Stefan Tsai example is a striking reminder of the fact that the vectors really change as a function of climate. It's the fact that currently there is spread of new vectors, but the viability of many of these vectors will also change as a function of time. And I think it just underscores the point that what you know or what we have been doing, if you just continue to do what we have been doing, it's not sufficient. And in fact, that is one of the trends when you see in that malaria line. Strategically, we are not doing anything different than we were doing 20 years ago or 15 years ago. And this is one of the problems. But the environments are changing so much more dramatically, and you might find that in some of those, it's valuable. We're going to do a class on by the numbers. You all will choose one problem area. It would be fantastic if one of you chose malaria and really dug much more deeply into these numbers, because it will be useful to figure out, is that surge from completely new regions? Is that surge from better diagnostics? Somebody giving an answer to why that uptick would actually be really, really fun to estimate. Okay, we're gonna run out of time. So William, maybe let's do a quick summary for schistosomiasis. So the
Perfect.
purpose of this is not the project that we are doing.
Mhm.
For the class, nobody's actually heard of I'm hoping nobody's gotten schistosomiasis. Some of you have heard of schistosomiasis.
Never had.
But nobody's had schistosomiasis. So just do a little touch on just the disease itself. And
Absolutely.
then in the very end, we can show people a full scope.
Absolutely. So I wanted to start by saying, has anyone heard of the idea of One Health? I love that that's being taught, and I've only heard about it in the last couple of years. So I think that's really important when we're talking about global health, to keep in mind that it's not just about human disease, but it's about ecological disturbances and the ways that we're actually directly, like we've been talking about, interacting with our environments, animals in our environments, plants, so on and so forth. And the reason I got into global health, I actually started in cancer neuroscience at the basic science lab and at the bench. And I was really curious the new toxicological exposures in our environment that might be contributing to cancer. We're seeing really concerning rates of different types of cancers. I want to be a pediatrician one day and see kids. And actually, after like leukemias and cancers of the blood, brain cancers are on the rise. And there's a need to understand what sort of environmental pollutants are really like leading to these neoplastic processes. And so this actually led me to talk to somebody who's a marine biologist, goes into the ocean all the time. His name is Dimitri Dehaene at UCSD, and he's one of the kind of premier microplastics researchers. And he found microplastics in his ocean samples on accident. So I wanted to start by saying, I love the microscope. I look at neurons all the time, but sometimes you find things on accident. And this is kind of a beautiful example. And this is one of our images, but he had an image pretty similar from ocean water that he had filtered, where there was autofluorescence under the microscope at immunofluorescing invertebrates, which is unrelated, but he was looking for a fluorescent signal, and he saw this. And he said, oh, this can't be right. It must be a scratch on the lens or something. Turns out, kept seeing it more and more and more over the years, and he realized this is something that's in the water. And so they did a pretty robust study and found that there's increasing amounts of microplastics right over in San Diego. And it turns out they've tried the same methods around the world, and they're actually contributing in NOAA, which is now, of course, falling apart with funding, but they've been trying to figure out where these plastics are going. So we got together with Dimitri, and this is my first global health project, was to go into Mexico and go to high altitudes, go to low altitudes, and sample the air, actually. And this isn't from ocean water, this is from my breath. So this was air samples as we were climbing a mountain. Turns out these microplastics are getting aerosolized and they're getting into the water cycle. So it's crazy. And this is like something that people aren't about enough, but you can learn a lot about the environment around you through the microscope. And this is kind of what got me into this world and led me to this global health challenge. So I wanted to give that little bit of
Oh,
background.
I love that. Yeah, and I
But
think microplastics is going to be a big section on when we start talking about the oceans.
it all comes back to the ocean or water sources. And this is a perfect example. So schistosomiasis or bilharzia is a neglected tropical disease that affects over 200 million people. Remarkable. I never heard about it until about 3 years ago. So what is it? It's a complex life cycle, but essentially imagine you're going into the water, like fresh water, to either swim or to fish or to do your laundry, and you can get directly infected through your skin by a tiny little worm that can then swim through your blood, and it has this life cycle of going to different organ systems. So if this is you going to the water and getting the infection, it has this life different organs, actually moves to the lungs, the liver, and then it starts to shed in the bladder system. And I want to show an area of the world where
And
this is.
we have schistosomiasis in the lab. We're the only lab in Stanford who keeps live schistosomiasis, if people want to see it. We keep the snails, and we've been doing this cycle. And one of the big reasons we can maintain schisto in the lab is because of a resource center that NIH runs, whose funding is also being cut. It's the Resource Center for Schistosomiasis, because they maintain live strains and maintain many of the life cycles and many of the snail species that transmit it. But downstairs, if people end up picking up schistosomiasis as one of the threads, we can actually meet this person or meet this entity.
In there, and it's something I should have mentioned. There's two main hosts. So you have a snail in the fresh water that's carrying this disease. And the other cycle is union host. And so there's several species that actually affect humans, and you then become part of the life cycle. So in order for this to perpetuate, you need both. And this actually contributes to like research challenges because sometimes you need like the snail host and you also need a mammalian host. But in the real life, it's basically it's a perfect example of this One Health concept we were talking about, because basically it's something that's both in the environment, in the snails, and it's in us. And so here in Nigeria, we've been working with collaborators there to study this, but in Southwest Nigeria in particular, there's very high prevalence and has been historically. It turns out this is because of places where there's dams and rivers. So another example of ecological disturbance, if you have a river that's dammed, you're interrupting the life cycle of a predator of the snail, which is freshwater prawn, and this allows the snail to then proliferate. And in these areas where there's dams, there's been a Lancet study where there's much higher rates of schistosomiasis. So unfortunately, what does this mean for people living there? At the Oyan River Reservoir, which is in Southwest Nigeria, everybody fishes, like every day, it's part of your occupation. So you are going to put yourself directly into the water, often just like getting in and out of the boat or just fishing. You're going to be touching the water, and if the route of exposure is through the skin, you can imagine how easy it is to get infected. And also it goes, so here's more about like the medical side of things. It goes infected fairly easily. There's a lot of illnesses that present like bloody urine, and that's the way you would see this if you're a healthcare worker or just a family member seeing, a patient who's concerned about bloody urine. There's a lot of things that can cause bloody urine or just pain while urinating. So this life cycle is
So just a second.
concerning.
How many of you have to leave right away? Yeah, so people who have to leave right away, that's good. We will record this, but we'll continue. Who can stay? Some of you can stay, maybe another 10 minutes. Okay, so don't worry about it if you have to leave. Yeah.
Thanks it's great to meet you. I'll see you soon.
Yeah, so on bloody urine, the story, a lot of people hear this story often enough out in the field, that a parent will bring their son to a clinic saying that my son is not menstruating. Because around that age, when you start getting these symptoms of bloody urine, every kid, and actually many in the sets of communities that we've studied, sometimes the number of infection can be 90 to 95% of kids in a class are infected. every single kid was peeing blood. So parents thought that this is the equivalent of menstruation. And so the one that was not peeing blood was brought to the clinic saying something is wrong with them.
Wow.
And sort of starts thinking about that when environmental exposure happens, all of the mass drug administration that's been in play, which is also a question right now, people are talking about how effective it is and should be continued. There is a drug that's been used for the longest time. It cures you, but the next month, next 2 months, you get infected again. So again, another type of a challenge in which you can't just say we cured it by finding a drug.
Right.
Because that's not sufficient because of reinfection. And so this is really why when we say One Health, it becomes important because the complexity of these diseases is such that unless it's eliminated, and then a human that's infected can have it for 30 years. So you could be perfectly healthy walking around, but you are shedding schistosomiasis, and only at 30 years or so, then you start internally bleeding and then it becomes completely out. But as a source, you can have even a single person in the community massively shedding egg. And then even in the snail, there is a 1 to million transmission there. There's the amplification factor, because when it grows in the snail, then it will multiply to release a massive number of cercaria every single day. So a human is releasing 30 to 40 eggs a day. There is another amplification factor that's at play. We have some eggs we can pass around.
Yeah, these are, say this is not going to infect you. These are fixed.
Yes.
And also they would need to infect a snail. So
That's correct. So
you can
this
just
is
check it out.
literally the safest sample that we can pass around. But it's valuable.
Yeah.
This is what you would be peeing out.
And it's fascinating because basically, if you think about the places where this would be a problem, like where would you go to the restroom if you were about to go to the restroom? You would have somewhere where you have a toilet that flushes it. You wouldn't have to worry about that getting into the water source where you're fishing necessarily, but that's not a reality. And that's something you have to keep in mind is like reconfiguring your daily habits and what would be going on. Like you'd be washing your clothes, relying on this water source that's actually infecting you. And so, like Manu said, the current like strategy, which kind of makes sense conceptually, is to give everybody this drug that cures the disease, right? Praziquantel treats the illness, but you're gonna just go to the water again. And if unless we figure out the upstream environmental effects or figure out a different strategy, there's a guy at Bay working on a vaccines. That's one idea. It's very far away. Another idea is some really cool stuff I'll hopefully get to talk about in a second, which is upstream environmental sources. But another idea which the WHO currently recommends is to have better low-cost diagnostics to test, treat, to try to work on, like Manu was saying earlier with malaria, like how does elimination actually work? Try to get it very low in the community. But again, if we're not thinking about environmental causes, it wouldn't work. To talk about some of the complications and why it's so concerning, if anyone's familiar with cervical cancer and how HPV infection leads to this, it actually, unfortunately, puts you at higher risk of that. So it puts you at higher risk of cervical cancer, which is deadly. Bladder cancer is there's a direct relationship there with the development of bladder cancer and infertility. And so we have a little comment on that, but I wanted to move along to, we talked to doctors in this area, which is just one of the areas, like we talked to them about what are the current challenges. And Dr. Adebayo there, listed at the beginning there, Dr. Adebayo told us that basically right now, screening efforts can look like carrying a generator out to community with a microscope, with a table, most of the time on bicycle with backpacks, and then actually setting up a transient clinic and then having to collect all the urine, put them on ice packs, bike city, and then get them to a lab where you can then centrifuge the samples. And I talked to Jim and Manu about transporting urine samples. I believe you were sharing a story from Senegal, or maybe it was from Kenya, but about the challenges of getting urine to somewhere you can actually take it to a lab. So this is when Frugal Science can maybe serve this purpose of how can you get portable and easy to use diagnostics. And I won't give a demo right now, even though I'm really dying to for your time, but this is Manu's creation, which is a portable microscope. And so, the whole idea of you take something that's mass produced, maybe have the minimal amount of dispensable supplies, and then be able to detect the illness would be helpful. And that's where, you know, we got excited about this project and started trying to work on
I think
this.
one comment that I'll make is, what's fun is, Will gathered a team, so there's a group of students, and I wanted him to just, because I spent a few days at Stanford, it just so happens that that day is today. And I wanted him to come here, because it's a great example of what a project can actually look like. You know, there are aspects of understanding the problem, there's aspects of a platform that already exists, or sets of platforms to develop. But the most important aspect is gathering the kind of information that's needed to really say, the approach that we have does make sense. The trick when everybody's selecting and choosing such a project is that it's driven by willpower, which Will has quite a lot of. That's important to have because you really do want to push any number of people to align around. Lots of people are there to help and support, but it's important to have that in the core team itself. And then simultaneously have this notion of that the mentors hang out and then the other teams and support infrastructure that we put around is driven by just like, oh, why don't I found this website, why don't I just have a call with this one person and say, hey, what is the worst they can say? They can say, no, I don't want to talk to you. That's not too bad. You are where you were before. So I think the purpose of this was not so much on Schisto because we will dive quite a lot in Schisto at a later time. But the purpose of it was to just understand that as Will is describing this, is all of these sets of things he's had to learn by just talking. And phone is still, phone and email, yes, but phone is still one of the most powerful underutilized technology. That just being able to be that resourceful around the ideas that you care about is actually important. That's the only way we connected.
Yeah.
Because I saw this message and just sat down and now they're here.
Yeah. So we're really glad to be here. Thank you.
Yes, I'm happy to.
And
get at least a day together.
Yes, it's incredible.
I leave to.
You should ask people, like send emails and try to, because we need creative thoughts and we need to see what's already being done and build on existing work. So that's kind of what we're working on. And I'll be bugging you all about this. But I wanted to also show like maybe an encouraging last thing. And this is how well you can see the eggs. It's really remarkable. We did an early experiment. And honestly, these are people that never touched the foldscope before. And also we made sure they'd never had microscope experience. That's amazing. So I'm convinced that sustainable development goals and should be used. We're talking to collaborators in Nigeria, but also I wanted to tell you guys, we've been getting messages from people in Tanzania who want to try it themselves. And I was like, we can share protocols, we can share all the wisdom and knowledge we've been learning from you. And that's the goal. It's like, can you make something scalable you can communicate with others about that can be done elsewhere? Because this, global health problems are global. Also, that mentor I wanted to tell you about with microplastics, he was doing field research in South Africa. So this is like a global problem. He's in San Diego, but he has schistosomiasis. So yeah, those are some of the next steps, but I wanted to briefly mention like encouraging upstream cause that people are working on really hard. Chelsea Wood, some people at Stanford and at WashU have worked on mapping the actual snail habitat. And they've actually tried, and we saw that snail habitat was where we were trying to work. Oh, and I actually don't have a slide on it, but they tried removing those plants, which are invasive species, hornwort plants, and they were able to use it for agricultural feed. So there's sometimes a solution that might have.
The purpose of that is to reduce snail population, which is a necessity.
Yeah.
Because most snails live on the vegetation. Ironically, there is the other plant side of the story. In Ethiopia, there is another plant that it hasn't been studied as yet, but there is some reports of a plant that makes a soapy compound. That when that plant is, you actually get reduction.
Wow.
There is also a rotifer that somebody discovered, which again is a beautiful aspect of thinking outside the box, that makes a compound that kills this infection in the snail, because the rotifer is in a symbiotic relationship with the snail and wants to keep the snail healthy. So the rotifer, which is small scale, 200, 300 micron, microorganism that lives in the vicinity of the snail is making a drug So a group at UCI just quantified what that molecule is. And now that molecule treats schisto inside the snail. It's a really beautiful story of thinking about kind of the symbiosis of where the sets of solutions come from in this One Health scenario are very ecologically, you have to understand the ecology. You have down. Even when you're working on a clinical problem, if you don't understand the broader perspective, you might miss out on many solutions that are not so obvious. So I'll send you that paper actually, just in terms of the ecological map side of the story. And Julio here at Stanford has a massive amount of work on the map.
Wow, I would
all the
love to
time.
talk to him.
Yeah.
So we talked to a drone operator.
Today, let's ping him.
That would be great.
Yeah,
So,
because we have a meeting today, minutes now at 12:30, on
shoot.
mapping measuring the planet. It might be that actually, we'll meet to talk. Okay,
Cool.
so I think this is the end of the class today.
Thank you guys.
Fortunately, so thank you, Will, for doing this.
Yeah, thank you for having